Thank you very much. I want to thank the organizers for inviting me. I wanted to come over to Florence, but there was a delay in my visa, so I could not come. But I'm happy to take this, give this talk virtually. The title of my talk is Epigenetics and Psychiatry. My talk will be somewhat general in nature compared to many of the previous talks. The topics I'm going to cover are classification of psychiatric disorders, outline of history of epigenetics and psychiatry, potential role of epigenetics in psychiatric disorders, dual role of genetics and epigenetics in psychiatric disorders, current status of epigenetics in psychiatric disorders, clinical implications, transgenerational inheritance, and taking epigenetics and psychiatry forward. Since this is a general audience, I will first briefly talk about the classification of psychiatric disorders. One group are the psychosis. These are severe disorders where there is marked impairment of behavior and inability to think coherently and comprehend reality. Clinical features of psychosis include delusions, hallucinations, and disorganized thinking. Examples are schizophrenia, abbreviated here as SZ, and bipolar disorder, abbreviated here as BD. Another group of psychiatric disorders are the mood disorders. These are characterized by changes in mood. They include major depressive disorder, here abbreviated as MDD, and it's also called major depression. Then there are the cognitive disorders, which present with confusion, disorientation, and memory disturbances. An example is Alzheimer's dementia, abbreviated as AD. Another group are the neuroses. Here the patient can comprehend reality, but suffering and disability may be severe in patients with neurosis. An example for neurosis is anxiety disorder. Then there is the child psychiatric disorders, which refers to those disorders that manifest during childhood. They include disorders like intellectual disability, attention deficit hyperactivity disorder, and autism spectrum disorders. Let me now go on to briefly about outline of history of epigenetics and psychiatry. The European-born American psychologist Erik Erikson propounded his epigenetic principle in the book Childhood and Society in the year 1950. Erikson proposed the psychosocial theory of development, describing important steps in an individual's relationship with others during his or her life. Later on, in 1982, the American psychiatric geneticist Irving Gottman and his colleagues published an influential book entitled Schizophrenia, the Epigenetic Puzzle. The author suggested in this book that since schizophrenia is a common disorder, it is likely to be an epigenetic puzzle involving genes and the environment in its pathogenesis. The molecular basis for Fragile X syndrome was found by positional cloning in 1991. Yuta Zogby's group discovered in 1999 that Rett syndrome is caused mainly by mutations in the MBCP2 gene. In 2004, there was a study by Meany, Ziff, and their colleagues, and in fact, Dr. Ziff is present in this conference. They showed that maternal behavior in rats, like licking, grooming, and arched back nursing, alters the epigenome of pups at the glucocorticoid receptor gene promoter in the hippocampus. The researchers also showed that these changes can be reversed by central infusion of a histone deacetylase inhibitor. Later, Meany, Ziff, Turecki, and colleagues translated these results to humans in a study on post-mortem hippocampus obtained from suicide victims with a history of childhood abuse. Since around 2005, that's since around the middle of the first decade of this century, epigenetic changes like DNA methylation, histone modifications, and non-coding RNAs have been increasingly investigated in psychiatric disorders. There's no let-up to the number of papers being published on psychiatric epigenetics. In fact, the numbers are only increasing. These studies have been done on post-mortem brain tissues of psychiatric patients and peripheral tissues on living patients. There are many difficulties associated with these studies. Firstly, it is not clear whether some of the epigenetic changes observed are the cause or the consequence of the disease process. Secondly, there's a lot of variability in epigenetic patterns within an individual, between individuals, and between study cohorts. Going on, epigenetic patterns vary between tissues and between cell types in the same tissue. Then epigenetic patterns vary with age in an individual. The living brain is the ideal tissue for epigenetic studies, but it is difficult to access. Going on, experimental conditions used by researchers influence results. Variables like addictive drugs and psychotropic drugs can affect the results. Having said that, a lot of work has been done, and now I'll go on to some new developments in the study of epigenetic changes in psychiatry. Now, epigenome-wide association studies, or EWAS, are being performed. New techniques and technologies like single-cell RNA sequencing, single-cell transcriptomics and epigenomics, and gene-specific editing are being employed. So what is the potential role of epigenetics in psychiatric disorders? Many lines of evidence suggest a role for epigenetics in psychiatric disorders. Firstly, the human brain is extremely complex, and the genetic information available is not adequate to specify all the neuronal interconnections in the human brain. Hence, additional information in the form of epigenetics is required to activate and modulate specific genes during brain development. Going on, epigenetics is thought to have played a major role in the evolution of the human brain. Then, among all human organs, epigenetics plays the greatest role in the development and functioning of the brain. In many psychiatric disorders, especially the neurosis, like anxiety disorder, environmental factors play a marked role in disease pathogenesis. As has already been pointed out in this conference, environmental factors more easily change epigenetic mechanisms of gene expression than the DNA sequence of genes. Going on, there's a low level of another factor pointing toward the role of epigenetics in psychiatric disorders is the low level of concordance in monozygotic twins for many of the disorders. For example, the concordance rate for schizophrenia in monozygotic twins is about 50%. Many psychiatric disorders like schizophrenia and bipolar disorder typically start relatively late in life. If they were genetic, why don't they start earlier, during childhood, for example, at the age of 3 or 4 or 5? There are differences in clinical manifestations between the sexes in many disorders. And this topic will be of interest to the attendees of this conference. For example, the onset of schizophrenia is earlier in men than women. Major depressive disorder is about 2 times more common in women than in men. Again, pointing to the possible role of epigenetic factors. Going on, there are fluctuations in the course of illness in patients with psychiatric disorders. For example, in bipolar disorder, there are swings from mania to depression. In patients with schizophrenia, there are times of remission and times of exacerbations. In some psychiatric disorders, parent of origin effects have been reported. For example, parent of origin effects have been reported in ADHD and late onset Alzheimer's dementia. But of course, more work has to be done in this particular area. Many psychotic disorders, like schizophrenia, are associated with reduced fertility of affected patients. But such disorders have not been eliminated from the human population.
That's interesting.
Why is that? Because schizophrenia there's a marked reduction in fecundity and fertility. Then how come schizophrenia has not been eliminated from mankind, from humankind? One possible explanation for this is that there's an interaction of genetic, epigenetic, and environmental factors for the development of schizophrenia. Now, going on to my next topic, dual role of genetics and epigenetics in psychiatric disorders. Genetics and epigenetics are closely related concepts. Genetic variations, this has already been pointed out in this conference, genetic variations can cause epigenetic variations. Conversely, epigenetic variations can cause genetic variations. Hence, it is not possible to discuss the epigenetics of psychiatric disorders without discussing their genetics. And briefly about the recent developments in psychiatric genetics. The field of psychiatric genetics is growing rapidly. The reason is there have been many technological advances and large international collaborations over the past decade which are enabling the genetic investigation of psychiatric disorders. GWAS are showing that genes are associated with psychiatric disorders like schizophrenia and mood disorders. But determining the ways by which associated genetic loci alter gene expression is proving to be difficult. Sequencing studies of disorders are also being done, and they're throwing light on the genetics. Such studies include whole exome sequencing and whole genome sequencing. Now, what are the strategies for translating GWAS data into disease mechanisms? Such strategies include the following: determining expression quantitative trait loci, or EQ to EL, which identify individual genetic variants with changes in gene expression. Next, determining methylation quantitative trait loci, or MQTL, which are genetic variants that can affect the DNA methylation patterns of CPG sites. Determining Mendelian randomization, in which a genetic variation is used as an instrumental variable to test for the causative effect of gene expression on the phenotype. There's another strategy, namely determining PRS, or polygenic risk score, which has been referred to in this conference previously. This may be a good measure to summarize the aggregate effect of pathogenic genetic variants. Another strategy is human-induced pluripotent stem cell-based models and CRISPR-based screens, which can dissect the pathophysiology associated with genetic variants. Consortia have been formed to elucidate the effects of variants found in GWAS studies. Such consortia include the Common Mind Consortium, the NIH Roadmap Epigenetics Consortium, and the Encyclopedia of DNA Elements Consortium. Now, what is the current status of molecular data in psychiatric epigenetics? There's a lot of data in this field, but there are some ambiguities and discrepancies. I will just refer to some of the findings in psychiatric epigenetics. The role of epigenetics in many child psychiatric disorders has been found out. Fragile X syndrome is due to TGG copy number expansion in the FMR1 gene, leading to FMR1 gene silencing. The silencing can be due to complex epigenetic changes like DNA methylation, histone deacetylation, reduced methylation of lysine 4, and increased methylation of lysine 9 on histone H3. These changes promote heterochromatin formation, which inhibits gene expression. As mentioned before in my talk, the majority of Rett syndrome cases are due to mutations in the MBCP2 gene. Molecular genetic tests are being used in the clinic to diagnose Fragile X syndrome and Rett syndrome. Going on, abnormalities of polychrome group proteins, or PCG proteins, have been implicated in the pathogenesis of disorders like Fragile X syndrome and autism spectrum disorders. In autism spectrum disorders, DNA methylation in the exon 1 area of the oxytocin receptor gene has been shown to correlate with social responsiveness deficits. In the psychosis, like schizophrenia and bipolar disorder, several abnormalities have been detected. Hypermethylation of the genes encoding relin, that is the relin gene, and the gene encoding GAD67, that is the GAD1 gene, has been found. In the frontal cortex of patients with schizophrenia, increased expression of HDAC1 has been found. In bipolar disorder and major depression, the gene encoding BDNF has been found to be hypermethylated. In major depression, the gene encoding the third serotonin transporter has been found to be hypermethylated. Many non-coding RNAs, particularly microRNAs, have been found to be dysregulated in schizophrenia, bipolar disorder, and major depressive disorder. 5-methylcytosine can be oxidized to another modified base, namely 5-hydroxymethylcytosine. And this modified base is environment sensitive. It is stable and highly enriched in the brain. Abnormalities in 5-HMC are being found in patients with psychiatric disorders. Histone methylation in the nucleus accumbens of mice has been found to play a role in increased vulnerability to stress caused by cocaine abuse. Anomalies of DNA methylation in stress-responsive genes have been found in patients with alcohol use disorder. Going on to the next topic, environmental factors and epigenetics and psychiatry. First of all, the factors during prenatal life and childhood. Some of these factors are of interest to this audience. Maternal stress which has been discussed previously, like anxiety and depression, obstetric complications, that is birth trauma, birth complications, early life trauma, for example, child abuse, poverty, diet, urban life. These are some of the early factors that can affect the propensity to psychiatry in an individual. Then during adulthood, there can be psychosocial problems like workplace problems, family problems, poverty, urban life, abuse of drugs, diet, chemicals, and migration. Going on to the next topic, clinical implications of epigenetics and psychiatry. First of all, biomarkers. Right now, there are no proper biomarkers to diagnose many psychiatric disorders, especially the disorders like schizophrenia, major depression, and bipolar disorder. And the epigenetic findings may provide the much wanted and much needed biomarkers to diagnose these disorders. But there are disparities in the findings from different studies on epigenetics and psychiatry. And what is the solution from this problem? One possible solution is to use a systems or network biology approach for developing biomarkers. Multi-omics data can help in this matter. Epigenetic data can be combined with genetic, clinical, transcriptomic, and proteomic data. This diagram shows how a systems biology approach can be used for identifying biomarkers. As shown on the left-hand side, multiple types of data can be, from multiple types of data, network models can be made. This can be fed into a computer, and from the computer, it is possible, at least theoretically, to differentiate patients from normal individuals. Such an approach may enable the use of personalized medicine for individual patients. It may allow use of biomarkers to assess clinical response to drug therapy, psychotherapy, and electroconvulsive therapy. All these therapeutic modalities alter epigenetic mechanisms of gene expression. So that is one way by which epigenetics may help in psychiatry. Second way is by epigenetic drugs. This also has been referred to in this conference. Epigenetic drugs may fulfill the long-felt need for new drugs in psychiatry. The drugs used today are very similar to those used in the 1960s. Most of the currently used drugs were discovered by serendipity or luck. Epigenetics may fulfill the need for rational drug design in psychiatry. Many of the existing drugs have in fact been shown to alter epigenetic mechanisms of gene expression. For example, it has been this conference has been said that clozapine can affect epigenetics. Other antipsychotics, in addition to clozapine, can also affect epigenetic mechanisms. When antidepressants can affect epigenetic mechanisms of gene expression, some of the mood stabilizers. But epigenetic changes are not the main effects of these drugs and may only contribute partly to their therapeutic efficacy. Some epigenetic drugs are in fact showing therapeutic potential. But as has also been pointed out yesterday, these drugs are pleiotropic. They have many effects. And they have other effects which should also contribute to their therapeutic effects. The methyl donor L-methyl folate is showing favorable results in the treatment of major depression. L-methyl folate is a methyl donor like SAM or SAMe. The HDAC inhibitor varinostat is undergoing phase 1 trials in Alzheimer's dementia. Dafidemostat, a histone 3 lysine 4 demethylase inhibitor, is going through phase 2b trials in schizophrenia and borderline personality disorder. Going on to the next topic, which will again be of interest to this audience, transgenerational inheritance of epigenetics in psychiatry. At present, there is evidence for transmission of epigenetic mechanisms in relation to psychiatry in animal models. In mice, it has been shown that anxiety and or social dysfunction depend not only on specific genetic alleles from one's parents and one's own experiences, but also on the experience of one's parents when they were young. So this is in animal models like mice. Little work has been done on the transgenerational transmission of epigenetic mechanisms in psychiatry in humans. In Holocaust survivors, and this will be of interest to some of the members of this audience, it has been shown that epigenetic alterations due to preconception parental trauma are present in both parents and offspring. However, it is difficult to eliminate the confounding effects of parental behavioral changes in such studies. It is almost impossible to rule out induction of these changes due to parental behavioral changes. So even after conception, after birth, the parents' behavior might change. How do you account for that? It's very difficult. In humans, it's very difficult to study. And to date, a conclusive study on transgenerational epigenetic effects on psychiatric disease risk has not been done. Taking epigenetics and psychiatry forward, I'm coming to the end of my talk. Lee and Avramopoulos have recently suggested the following steps to take epigenetics and psychiatry forward. Basic research, further elucidation of epigenetic enzymes and pathways, functional characterization of locus-specific epigenetic changes, improvements in the enrichment of homogeneous cellular populations, translational research, development of transgenic and knockout animal models, site-specific manipulation of the epigenome, study of effects of drugs and the environment on the epigenome, research in humans, epigenomic studies on post-mortem brains. Right now, epigenomic studies, that is across the genome, are mainly being done on peripheral tissues. But it will be good to study this on post-mortem tissues. Then correlation of epigenetic effects between tissues. Identification of peripheral biomarkers for psychiatric disorders. Integration between genetics and epigenetics. And research strategies to establish norms to facilitate integration of data sets from different studies. To conclude my talk, there is increasing evidence that epigenetic dysregulation contributes to a broad spectrum of psychiatric disorders. Epigenetics is a link between environmental factors and the genome in psychiatric disorders. A clearer understanding of epigenetics and psychiatry may pave the way for new biomarkers and drugs for these disorders. Acknowledgements, I acknowledge Dennis Grayson, Dimitrios Avramopoulos, Dag Yasui, and Trigvi Tolasball, with whom I have collaborated. These are some of the selected references. Thank you for your patience.